Many thanks for the opportunity to speak today. I'll be speaking on 30 years of PET. Where are we now? An era of innovation, impact, and opportunity. I'll be covering novel traces, the expanding role of PET, particularly in theranostics, new technology, collaborative clinical trials, guidelines and leadership, the potential for AI in imaging, and precision medicine. FDG has been the mainstay of clinical PET in Australia for 3 decades, and oncology remains the main indication for FDG PET imaging. However, the indications for FDG are broadening. We've recently had funding through MBS for Alzheimer's evaluation of FDG PET brain imaging. FDG PET's also playing a role in cardiology, particularly an emerging role in looking at cardiac myocardial inflammation, such as with sarcoidosis, and also a role in cardiac viability. And FDG PET is actually very promising in inflammatory conditions, and we're probably just starting to explore those indications, such as for pyrexia of unknown origin, particularly with staph sepsis, vasculitis and inflammatory conditions, and atherosclerosis. But there has been the emergence of new PET tracers over the last few years, which have evolved from the research domain to clinical use, such as gallium-68 octreotide PET in neuroendocrine tumours, which was Medicare funded from 2018, and PSMA PET in prostate cancer, which will be Medicare funded from the 1st of July this year. So these are our clinical oncology currently funded PET tracers, including FDG PET, gallium-68 octreotide PET for neuroendocrine tumours, and PSMA PET in prostate cancer. But there's a number of other PET tracers which are used quite often to answer clinical questions that are often used in our clinical care but are not currently funded. Sodium fluoride PET was used moderately extensively in Australia, and in fact was a substitution item when we had issues with technetium availability. And sodium fluoride PET provides really exquisite imaging of the skeletal system and is a very valuable bone imaging agent. I would say in Australia we probably underutilise sodium fluoride PET, and that's likely because it is not currently funded. This is an image that we undertook at our site when we had issues accessing technetium bone scans, and this was a sodium fluoride PET scan performed in a patient for cancer evaluation. The amino acid PET agents are showing great promise in brain tumours, and there's currently a study of PET in Australia, and this is actually an important and emerging area. There's a lot of interest in iodine-124 PET imaging, both as iodine-124 by itself for advanced thyroid cancer, but also labelled to agents such as MIBG for whole body MIBG PET imaging. Similarly with F-DOPA PET, it has indications in paraganglioma and pheochromocytoma in the whole body imaging domain. In the research domain, there are more emerging PET tracers. FAPI hit the world by storm in the 2019 SNMMI Image of the Year, as it appears to have widespread applicability across a number of different cancer types and looks like a really promising imaging agent with high target to background ratios. There's also the emergence of the antibody imaging and using the longer half-life imaging agents such as zirconium-89, and this example with zirconium-89 to label an anti-PD-L1 antibody to look at immuno-PET imaging and to ascertain if this corresponds to response. And I think we will start to see studies such as these more and more emerging in the PET domain. In neurology, we have FDG PET. As I say, it is now MBS approved for the evaluation of Alzheimer's disease and also for brain tumours. Amyloid PET has an important role, particularly for a selection of patients for clinical trials, but is not currently funded in Australia. And F-DOPA PET is really valuable in some of the complex movement disorder patients. In this case, it's an example of Parkinson's disease with a very typical asymmetric posterior putamen reduction in F-DOPA activity. And these are all important PET tracers which are beyond FDG as well. So PET only not has an increasing breadth in terms of the traces, there's also an expanding role of PET, and this particularly is in the area of theranostics. And theranostics, as everyone is aware, is a combination of diagnostics and therapy based on the principle of performing a diagnostic test which will determine a personalised treatment. So in nuclear medicine, we usually use the term theranostics to a diagnostic therapeutic isotope pair, such as gallium PSMA for PET imaging in this case, and lutetium PSMA for therapy. And this SNMMI Image of the Year from 2018 from Michael Hoffman and his group at the Peter Mac shows really impressively how the PET imaging showing the target of PSMA is showing pre and post therapy in patients receiving lutetium PSMA therapy. So the PET imaging can be used to evaluate disease pre and post therapy and also to evaluate target. And in this case, very impressively shows some of the early results of lutetium PSMA therapy. And we see this in prostate cancer, and many of us that are now utilising lutetium PSMA therapy are seeing that you can do a baseline PSMA PET scan showing quite extensive disease, and then following as little as 2 cycles of therapy, the PSMA PET scan can again be used to evaluate disease, and in many times this shows a really dramatic response to therapy. And so the PET imaging component of the theranostic treatment is very important for monitoring the disease. This is also the case in neuroendocrine tumours with gallium octreottate PET imaging. Again, this is a clinical example of a patient with a locally advanced unresectable neuroendocrine tumour treated with lutetium octreottate therapy, and the gallium-68 octreottate PET at baseline and post-treatment was very useful to evaluate the response to therapy. But importantly, PET can be used in a more sophisticated way, and it can be used to ascertain and evaluate patients for selection for these targeted therapies. And so you can perform a targeted scan, such as the PSMA PET scan, to evaluate whether the patient is suitable for a targeted therapy. And in this case, on the left, you can see a favourable distribution. There is very high expression of the target, which is PSMA. But when you combine it with an FDG PET scan, you can also do this to ascertain, to make sure that there is not a high volume of off-target disease that wouldn't be treated by targeted therapy. So when you see the unfavourable distribution example, you can see relatively low expression of PSMA on the gallium PSMA PET scan, and on the FDG PET you can see much more extensive disease and areas of mismatched disease. And if you're treating this patient with a PSMA targeted therapy alone, you might expect a much less likelihood of response because there are many sites of disease not expressing target. And so we're learning to use PET in a more sophisticated way by combining different traces to try and understand disease better and plan our treatments more effectively. And I would say in Australia, particularly in the theranostic domain, we've actually been really quite advanced in the way that we're utilising this. In PET, we now have some amazing advances in technology, and total body PET is one of these. And thanks very much to Steve Meikle for these slides, which I really appreciate, which demonstrate the total body PET. So with conventional PET, with the patient passing through the gantry, we're only gathering the positrons over a small field of view, and it's relatively inefficient. In a total body PET, where there are in fact crystal detectors all the way around the patient, you have a much higher sensitivity. And total body PET is providing amazing and wonderful new opportunities, and we really, I think, are all looking forward to seeing how this changes our practice. Total body PET has a much greater signal to noise ratio, so is much more sensitive for detection of disease, performs much faster scans, so you can get higher patient throughput and less chance of patients moving while they're having a scan. You can perform delayed imaging because you can have much lower count rates and still detect sites of disease. And there's a much lower dose or radiation dose to patients, so there might be the potential for expanded use in paediatrics or maternal fetal or studies of normal biology in patients or normal volunteers. Scientifically, the dynamic imaging aspects are fascinating, and I think what we might learn in pharmacokinetics will be incredibly interesting, particularly as we explore novel molecular targets, biomarkers, and new treatments. You can look at signaling between organs, and we can track targets over a long period of time. And really, I think the value of total body PET in research and clinical trials really looks fascinating. There's been the Explorer Consortium working on total body PET, and there are also now 2 commercial total body PET systems, the United Imaging Explorer and the Siemens Biograph Vision Quadra. I've taken these images actually from the Siemens website just to show. On the right is an image of a patient, and this was acquired of a 10-minute imaging time. But on the left, this is the same patient, but only a 1-minute acquisition time, which actually has given incredibly good quality images and really shows the potential of these systems. And I note that this month there is a hot topics out of the Journal of Nuclear Medicine on total body PET. Will it change science and practice? And in fact, this is an example given of a dynamic imaging set with a fluorinated fluoroglutamine showing the initial 2-second acquisition in a dynamic phase, a 15-second acquisition at 30 seconds showing the biodistribution, a 30-second acquisition just 2 minutes after administration, and then at 30 minutes, an acquired image. And really, what you can learn from these images in the biodistribution of new traces, disease, drug development, I think will be fascinating. It's an amazing advance. I just wanted to spend some time talking about collaborative multi-centre clinical trials in Australia. And ARTnet has been established since 2014 and has played an important role in facilitating multi-centre clinical trials throughout Australia. So ARTnet is a joint initiative of the ANZSNM and ANMS with a mission statement of promoting and facilitating innovative collaborative clinical research utilising radiopharmaceuticals for imaging or therapy. And ARTnet has a scientific committee who undertake the scientific activities such as review and endorsement of clinical trials, development of imaging and radiopharmaceutical manuals, in collaboration with the investigators of clinical trials. ARTnet has a camera credentialing program and a radiochemistry certification program, which are all activities to support multi-centre clinical trials within Australia. And the quality activities of ARTnet in clinical trials, the value of these was recognised very early on. And in our first prospective study looking at gallium-68 PSMA PET imaging as part of the Pro-PSMA study, we had a very unexpected finding, and that was a 15% SUV variation when we assessed different camera systems across Australia using a phantom. Ultimately, this was found to be a dose calibrator, calibration error. But had we not undertaken the study, the phantom study across multiple sites in Australia, we would have really had very unreliable results when we tried to combine imaging results across trials. And a 15% SUV variation is very significant. So thanks to Dale and the team of people involved who did a lot of hard work to find out where the error was coming from and to ensure the quality of these clinical trials. So performing multi-centre clinical trials in Australia, for which PET is actually very integral to these, has been something for which ARTnet has helped to assist with and facilitate to provide that opportunity. And this is an image actually from Michael Hoffman. I think this one is from the therapy study showing the number of involved sites in a study, which was 11 sites in the therapy trial all across Australia. And so sites are collaborating across Australia, but there's also been very strong collaboration with a number of different clinical trials groups. And I think that's really strengthened the quality of research that's being performed in Australia. And this includes with ANZUP, TROG, COGNO, ALLG, and many other clinical trials groups for which there is very strong collaboration, which is important. So having multi-centre collaborative clinical trials in Australia has a number of advantages. This has allowed us to really accelerate in the way that we recruit patients to studies in a much greater way than could be achieved in a single site. This was demonstrated in the Pro-PSMA study quite impressively, which actually, although 10 sites across Australia were required to be validated for this trial and follow the same protocol, it ended up recruiting 6 months ahead of target. Which just means that you can then evaluate and publish your evidence a lot faster and actually accelerate that development program. Similarly, with the therapy study, which was a therapeutic trial, again, 11 sites were required to be credentialed and opened across Australia, and that recruited way ahead of target as well, which was really incredibly impressive for quite a complex study. But importantly, doing multi-centre clinical trials in Australia has led us to developing evidence and providing impact. An example of this was the Gallium PSMA management intent study. This was actually published in 2018 by Paul Roach et al. And this looked at 431 patients who had PSMA PET imaging performed, 25% for primary staging and 75% in the biochemical relapse group. And clinicians were asked to indicate management pre and post the scan. And this study found that PSMA PET led to a change in planned management in 51% of patients overall, which was actually very high. This effect was even higher in the biochemical relapse group, of which 62% of patients, there was a change in management intent. And in the primary staging group, it was slightly lower at 21%. So this study coming out of Australia has had quite a lot of international impact. And last year, the Journal of Nuclear Medicine published just a short editorial talking about the impact factor, which has now increased to 10.057. And there were 10 papers that significantly contributed to the impact factor. One of these was, in fact, this management impact study from Australia. So it has been internationally highly cited because it showed how PSMA PET imaging actually practically changes what happens to patients and their management, and has been quite an important study and was very collaborative. Similarly, the Pro-PSMA study, so this was the study of PSMA PET imaging in high-risk prostate cancer patients before curative intent surgery or radiotherapy. This is a study which has also had significant impact both nationally and internationally. So this study, led by Michael Hoffman, was published in The Lancet in 2020. And PSMA PET, when compared to conventional imaging with bone scan and CT, the PSMA PET was found to have greater accuracy, more likely to result in management change, less radiation exposure, high reporter agreement, and less equivocal findings. This was shortly followed by a cost-benefit paper published in European Urology in 2021, showing that PSMA PET was also cost-effective when compared to bone scan CT. So why is this important? Well, evidence is important because it leads to outcomes, and it leads to outcomes which will ensure we can actually integrate these PET imaging agents into clinical care. So in Australia, there was an MSAC review of PSMA PET imaging undertaken, with MSAC recommending funding of PSMA PET imaging, both for primary staging and biochemical relapse. And recently, as tweeted here by ANMS, PSMA PET CT has actually been funded from the 1st of July 2022. So establishing evidence can lead to funding and integration into clinical practice, and also integration into clinical guidelines, where studies such as the Pro-PSMA trial form part of international guidelines for the care of patients with prostate cancer. The therapy study is another incredibly important and internationally impactful study. This was published in The Lancet in 2021 by Michael Hoffman et al., this study compared lutetium PSMA 617 to cabazitaxel in patients with advanced prostate cancer. And it demonstrated a high PSA reduction with lutetium PSMA, a favourable progression-free survival and objective response rate, and a relatively tolerable side effect profile. So this was a randomised, multi-centre prospective clinical trial, 11 sites in Australia, which has provided international evidence for the efficacy and tolerability of lutetium PSMA therapy. So there are a number of current open studies in Australia which ARTnet is supporting, including in prostate cancer, the ENZAPI upfront and evolution studies, which are combination therapies of lutetium PSMA, and also the primary 2 imaging study. In brain tumours, there is the FET glioma study, which is incredibly important for providing evidence for FET-PET in high-grade glioma. And shortly to commence is the iFirst study in thyroid cancer, and that's looking at iodine refractory advanced thyroid cancer and redifferentiation therapy. So these are all areas where it is important to gain evidence, and for which in Australia, by working collaboratively, we really are working towards this in a very effective way. I just wanted to touch on guidelines and leadership. There's been a number of guidelines and leadership for which Australia and our experience in PET and in nuclear medicine has had international impact, including the Lancet Oncology Commission of Medical Imaging and Nuclear Medicine in Oncology, guidelines in COVID-19, and guidelines such as joint EANM, SNMMI, and ANZ-SNM practice guidelines of FDG PET in immunomodulatory treatment. There has also been hot topics such as in ARTnet, and also looking at theranostics and the importance in training, accreditation and guidelines from Australia. So what we're doing in Australia in these particular areas has international impact, and we are internationally connected as well in many areas. And these are just only a few examples of the fantastic work that is occurring. I just wanted to touch on AI, or artificial intelligence, and the role of this in imaging. This is both something which is current, emerging, and is certainly an area of innovation. And this paper that recently came out in 2021 was looking at the number of publications of AI in imaging. And you can see this really becoming exponential. And we're starting to see this occurring in the research domain and starting more and more to enter the clinical domain as well. And there's an area that certainly for us in PET in this decade and beyond is something we will start to see more and more. This paper from 2019 looks for where AI might impact in our imaging workflows. And in fact, there are multiple areas for which AI could provide assistance in the workflows, from planning to scanning to reading to reporting. And we're really looking forward to how AI will improve our practice as we move forward. And lastly, I just wanted to speak about precision medicine and the role of PET in precision medicine, which is a role which I see as critically important. So PET imaging allows us to understand disease and guide therapy, which is really the very essence of precision medicine. Traditionally, when we've thought about cancer, we've thought about staging cancer in a TNM, tumour, lymph node and metastases way, very much based on anatomical staging systems and histological findings in terms of tumour infiltration locally. More and more it's recognised, though, that tumours are not just about the anatomical location, they're also about the molecular markers and molecular pathways within the tumours, which affect very much the prognosis and the treatment of patients and subsequent response. So characterising tumours will be not just about their anatomical or physical location in the body, but more and more about their molecular profile. And what does this mean with imaging? Well, this is a slide and an image, a figure that I often use. It is from a while ago, from 2010, but it shows a number of different molecular pathways in glioma. And when we look at PET imaging, because PET images molecular pathways, in this particular example, there's an example of FDG PET imaging, amino acid PET imaging, and FLT PET imaging. But there are multiple other molecular pathways which PET can be used to actually image, both as cell surface markers, intracellular processes, and also within the tumour microenvironment. And we have an opportunity with PET to be able to really understand biology and disease through imaging. And we need to always remember that what we're actually imaging is a biological process. So if we really want to understand disease, then in PET we have an amazing opportunity. We can combine both biology, science, molecular markers with the development of novel radiochemistry and radiopharmaceuticals. Our advances in imaging are allowing us to really push the field in what we can actually image, total body PET, allowing dynamic imaging, fantastic sensitivity. And then as we develop AI techniques, we will start to understand our imaging further. So I think we have this amazing and unique opportunity within PET imaging to really have an incredible role in precision medicine in what we do. And I recently saw this posted on LinkedIn and it caught my eye. And it caught my eye because of the headline, FDG is not a contrast agent. And this is absolutely true. FDG is not a contrast agent. FDG is a signaling pathway and it is biology and is helping us understand disease. And this publication that came out this month in the JNM is talking about how FDG is telling us about cellular processes, about how they differ in different tumour types, how they may affect prognosis, and how treatment may change the expression. So FDG is not a contrast agent, and what we do in nuclear medicine and PET imaging is all about the biology and understanding disease. So 30 years of PET in Australia. Well, this is 30 years of advancement of PET in Australia, and the current era is one of innovation, impact, and opportunity. Nuclear medicine in Australia is internationally recognised for clinical trials and evidence, quality activities and guidelines, education and training. And we are multidisciplinary and collaborative. And therefore, I think we're extremely strong and should be very proud of what we've been able to achieve. I hope that you can see from all of this that the future for us in nuclear medicine and in PET is incredibly promising and will provide more advances as we develop novel radiopharmaceuticals and those are integrated into clinical care pathways. There will be technology advances, including in molecular targets, imaging equipment, software, AI, and analytics. And the PET has an absolutely key role in precision medicine, which is imaging to understand disease and guide treatment. So I put the question out there, what will the future be in 2032 in PET in Australia? And I have to say, I myself feel the future is incredibly exciting and cannot wait to see what we achieve in the next decade. Thank you very much.